Hello and welcome everybody to my channel OmniBio Entrance, and today we will be discussing CSIR PYQs for the years 2012 and 2013. I will be discussing only the genetic section. If you are new to my channel, please do like, share and subscribe. So this is June 2012 question paper, CSIR Life Science. I am doing the genetics part and coming to the first question. How many genetically different gametes can be made by an individual of the genotype capital A, capital B, small b, small c, capital D, capital E, small e, assuming they are independently assorting. So they have asked for how many different gametes can be made by the individual for this particular genotype. So the number of gametes, it is given by the formula 2 raised to n, where n is the number of heterozygotes in the genotype. So here the number of heterozygote is 1, 2 and 3. So 2 raised to n. So 2 raised to 3. That is 8. So the answer here is option 3, 8. Then next one. When two independent pure lines of P with the white flowers are crossed, the F1 progeny has purple flowers. The F2 progeny obtained from selfing shows both purple and white flowers in a ratio of 9:7. The following conclusions were made. A similar question was asked in December 2017, I think. Anyway, when two independent pure lines of P with white flowers, so they both were white flowers. So we will take it as capital A, small b. Crossed with small a, capital B. Okay. Now upon crossing, you will get capital A, small a, capital B, small b. This is the F1 progeny and this is purple in color. So these two, that is capital A small b and small a small capital B, they are white colored flowers, and upon crossing you get purple colors, where the two dominant genes, that is capital A and capital B, they come together and form what is called as the purple colored plant. Now upon they have the F2 progeny obtained from selfing shows both purple and white flowers. F2 progeny is, this is F1 progeny. Capital A, capital B, small b is an F1 progeny. You self the F1 progeny. That means you cross with the another purple flower having the same gene, not another purple flower, with the same flower having the, of the same plant, having the same genotype, you cross them, and that is purple in crossed with purple flower. You get purple and white flowers in the ratio 9:7. Now this ratio shows that it is complementary gene action. Now we will come to the conclusions that were made. The two different genes are involved, mutations in which lead to formation of white flowers. That is correct, because here there are, if you see this one, capital A, small b, B is mutated. But when A got crossed with small a, capital B, A and B came together and it resulted in purple flowers. So both these two different genes, that is capital A and small b, and capital B are required for the formation of purple flowers, and in the presence of only one dominant gene you got white flowers. Okay, so two different genes are involved, capital A, capital B. Mutation in which lead to formation of white flowers. So if B is mutated or if A is mutated, it lead to the formation of white flowers. So first option A is correct. These two genes show independent assortment. That's also correct. This is an example of complementary gene action that I already told you. This is an example of duplicate genes. No, it is a complementary gene action. So the answer here is A, B and C are correct. Now next one. A cell undergoing meiosis produces 4 daughter cells, 2 of which are aneuploids, while 2 are haploid. This can occur due to. So they have told 2 of which are aneuploids. So here it is n plus 1, n minus 1, while 2 are haploid, n, n. So you have got 4 chromosomes. This is meiosis 1, the chromosomes separate. Then comes the separation of the chromatids that happens during meiosis 2. But here one of the chromosomes, that chromatids fail to separate. So there is non-disjunction during meiosis 2 if you see for the blue colored chromosome. But the red chromosome, it is perfectly normal during meiosis 2. So the blue color chromosome, you have got one extra chromosome along with this particular chromatid. So it is an aneuploid, it is n plus 1, and the other one doesn't have one particular chromatid. So it is n minus 1. Whereas the red chromosome, they both are perfectly normal. So n, that is haploids after meiosis 2. So they have asked non-disjunction during meiosis, meiotic division, first meiotic division only. That's not correct. So answer then non-disjunction during second meiotic division only. That is correct. Only then you will get aneuploids and haploids. So option 2 is the right answer. Then next one. 3 E. coli mutants are isolated which require compound A for their growth. The compounds B, C, D are known to be involved in the biosynthetic pathway to A. In order to determine the pathway, the mutants were grown in a medium supplemented with one of the compounds A to D. The results obtained are summarized below. So these are the results. Now if you see the table, if you see all plus, that is the required compound that has to be obtained from the pathway of B, C and D. So A is the required compound. So A has to come at the end. So option 4 can be deleted because A is coming in the beginning. Whereas B for 1, 2 and 3, A is coming in the end. So out of that, which one is the pathway for the production of A? Now if you see A is all plus. Now you see for a compound where there is a reduction from A plus, not A plus, there is a reduction in plus from when compared to A. So that you can see in D. Here there are 3 pluses and here only 2 pluses are there. So D should be the substrate for A. So before A, D has to come. Now there are 2 pluses in D. Then C for another compound where there is only 1 plus less than D. So that is C. So D, before D, C has to come. Then C for where there are all the values as 0. And that 0 value, it should come in the beginning. So B, C, D, A. That is the pathway for production of A. So that is how you see for this particular table and deduce the pathway. But if you see the other way around, okay, now suppose I am explaining everything. This is mutant 1. When A is added, it shows growth, but when B or C or D is added, it doesn't show growth, indicating lack of enzyme needed for the conversion of these compounds, that is B, C and D to A. Now if you take the case of mutant 2, here upon addition, when A is added or D is added, there is growth, so it has got the enzyme for converting D to A, but lack the enzyme needed for conversion of B to C. So it can and C to A. So from here we can deduce that D is directly getting converted to A. Now next one. Mutant 3. Now if you see the case of mutant 3, it shows growth when A, C or D is added because it has got the enzyme needed for conversion of C to A as well as D to A, but we already deduced that D is getting converted directly to A. So C has to get converted to D and then D has to get converted to A. So what is left out? B is left out. So B, C, D, A. So the answer here is option 1. To the next question. Following are 4 modes of inheritance: X-linked recessive, X-linked dominant, autosomal recessive, autosomal dominant. Which of the above modes of inheritance can explain the pedigree shown below? Now if you see the pedigree, you see that both the male and the female children are affected from the affected or deceased father. So it must be an autosomal condition. Now they have given A, C, C, D options, B, C, D. So if you see wherever A and B comes, that is X-linked dominant and X-linked recessive, you can easily tell those options are not correct. So option 1 is not correct, option 2 is not correct. Now comes the case of C, D or D only. So you have to see whether it is autosomal recessive, autosomal dominant or just autosomal dominant only. Now here is the conditions. Now if it is dominant, so this has to be capital A, small a, small a will be this, because this is not having the disease, whereas this is diseased and it is autosomal dominant. Now since there are daughter and this is daughter and the son which is affected with the disease, so naturally this has to be a heterozygote. That is why capital A, small a, small a, small a. So the daughter and son, they will be capital A, small a, capital A, small a. Now the daughter is affected here and this is the normal male. So he has to be capital A, small a, because this is a dominant condition, and then you will get capital A, small a, that is affected daughter and unaffected son. So that is the case of dominant autosomal dominant condition. Now coming to autosomal recessive. Now if you consider this as small a, this also small a, and even this also small a. Now if these two have to be small a, and the mother is unaffected, she has to be a heterozygote. So naturally you will get small a, capital A, small a, capital A, small a, and upon crossing capital A, small a with small a, you will get small a, and capital A, small a. So this is a case of autosomal recessive pedigree also. So answer is option 3, that is C and D, autosomal dominant and autosomal recessive. So the answer here is option 3. Now coming to the next question. The auxotrophic strains of E. coli A, that is met minus, bio minus, threonine plus, leucine plus and thymine plus, and B, that is met plus, bio plus and all the others minus, were incubated together for 18 hours in a liquid complete medium, and then approximately 10 raised to 8 cells were plated on a minimal medium. Prototrophs were observed at a frequency of 1 in 10 raised to minus 7 cells. Prototrophs means those which can synthesize their own, or I can say they can grow on minimal medium. So this may have happened by a process of genetic recombination between two strains or by mutation of the strain. So this arisal of this prototrophs could have been because of genetic recombination or because the two strains got mutated. Or by mutation of the strains. So now, which of the following control experiment would help rule out the possibility of mutation? Now you have to see for a control for this particular experiment which will help rule out the possibility of mutation. So you have conducted a mutation, an experiment where auxotrophic strains of E. coli A and B were mated by growing them for 18 hours in liquid complete medium, and out of that 10 raised to 8 cells were plated on minimal medium. Then you found out that there were prototrophs being observed on plating on minimal medium, and you are worried why you got prototrophs, whether it is because of genetic recombination between the two strains or is it due to mutation of the strain. So what you do is you have to set up a control for the experiment. Now which control experiment would help rule out the possibility of the mutation? So answer is option 3, that is growing strains A and B individually in a liquid medium for 18 hours, liquid complete medium. Growing strains A and B individually in a liquid complete medium for 18 hours and then plating them on minimal medium. So what will happen is, if it is due to mutation, if the prototrophs that you got upon incubation of the E. coli strain A and B together, if the prototrophs that you got, they have arisen due to mutation, then the prototrophs, then upon plating on minimal medium, you should have seen growth. Okay. Now what you do is, for getting the control, what you do is you grow the strains A and B individually in liquid complete medium. That is, A is grown in liquid individually in one conical flask, and then B is grown in another conical flask, and you don't allow the cells to undergo recombination. There is no conjugation between the cells because they are placed in separate conical flasks, and that too in a liquid complete medium. Then you incubate for 18 hours, and then what you do, then you plate them onto minimal medium. Now if the prototrophs have arisen due to mutation, then upon plating on minimal medium, you should have seen growth. But since you don't see growth, if you don't see growth, then the prototrophs have arisen because of genetic recombination between the two strains. Okay. Coming to December 2012 question paper, coming to the first question. A cross was made between pure line pure wild type male and brown-eyed curled-winged females of Drosophila melanogaster. The F1 females were test crossed. The F2 progeny obtained was as follows. So a cross was made between pure wild-type males and brown-eyed curled-winged females of Drosophila, and the F1 females that were obtained, they were test crossed, that is crossed with the recessive parent, and F2 progeny was obtained. So this is the value of F2 progeny. So wild type is 200, brown eyes curled wings is 150. So 200, 150, they are maximum value, so they are your parentals. Brown eyes, normal wings, normal eye, curled wings, they are your recombinants, 30 and 20. And this is your total number of progeny. So that they have asked for the genetic distance between brown eye and curled wing loci. So recombination frequency, that is number of recombinants by total number of progeny, is 30 plus 20 divided by 400 into 100. You will get it as 12.5. So the answer is option 1. Then next one. The effect of nonsense mutation could be nullified by reversion as well as suppression. Which of the following processes will help distinguish between the two kind of revertants? So it is recombination that will help to distinguish between the two kind of revertants. Then 2-aminopurine induces mutation by. So 2-aminopurine, it causes transition mutations. So it is not frame shift, neither insertion nor duplication, it is base pair change. Answer is option 1. The next one. The RFLP pattern observed for two pure parental lines P1 and P2 and their F1 progeny is represented below. Further, the P1 plants had red flowers while the P2 had white flowers. The F1 progeny was backcrossed to P2. The result obtained showing the number of progeny with red and white flowers and their RFLP pattern is also presented below. Now you see that the parentals, that is P1, pure parental lines, red and white. Red has got one single band, white has got two bands. Now this is the F1 progeny obtained upon crossing the two pure parental lines, that is P1 and P2. So red has got 3 bands. From both the parents combined, it has got the 3 bands. That is RFLP pattern. Then next what you did was you backcrossed the F1 with P2. So red was crossed with white or white was crossed with red. And what did you get? You got parentals, that is red, 3 bands, white, 2 bands. They are 45 each. They are your parentals. These are the recombinants, that is 5. Red 2 bands, white 3 bands, 5 each. So the DNA marker and the gene for our color, they are 10 centimorgan apart because the number of recombinant progeny is 5 each. So the answer is option 1. The next one. Wild type T4 bacteriophage can grow on B and K strains of E. coli forming small plaques. R2 mutants of T4 bacteriophage cannot grow on E. coli strain K, that is not permissive host, but forms large plaques on E. coli strain B, that is permissive host. The following 2 experiments were carried out. Okay, now wild type T4 bacteriophage, it can grow on B and K strain of E. coli and form small plaques. That is your wild type. It forms small plaques on both B and K strains of E. coli. Now R2 mutants of T4 bacteriophage. So there is a particular mutant variety for T4 bacteriophage, that is R2 mutant, and it cannot grow on E. coli strain K because it is a non-permissive host. but form large plaques on E. coli strain B. But the R2 mutant, it can grow on strain B. And they conducted following experiments. So experiment 1. E. coli K cells were simultaneously infected with two R2 mutants A minus B minus. So there are two R2 mutants, one is A minus and the other one is B minus. So here first mutant A is mutated, in the second one B is mutated. Several plaques with wild type morphology were obtained. Now they have already told that R2 mutants of bacteriophage cannot grow on strain E. coli K. But how come the several that when you infected with two R2 mutants, when E. coli K cells were simultaneously infected with two R2 mutants, how come you got plaques with wild type morphology? Now that may be because of a phenomenon called as complementation. That is, mutations A and B, they belong to different cistrons and they complement each other and plaques are formed. So A minus will have B plus, which is unmutated, and B minus strain it will have A plus, which is unmutated. Now A plus and B plus, when they came together and they complemented each other, resulting in the formation of wild type morphology. So the first experiment, it is due to complementation. Now experiment 2. E. coli B cells were simultaneously infected with the same mutants as above. Now B cells, they are permissive for mutated T4 bacteria, R2 mutants of T4 bacteriophage. T4 phages were isolated from the resulting plaques and used to infect E. coli K cells. Now you got so many T4 plaques from the E. coli B cells and they were used for infecting E. coli K cells, which is a non-permissive host. And what did you obtain? You got few plaques with wild type morphology. Only very few plaques were obtained. Now what could be the reason for this? The reason is recombination has taken place. This recombined gene, it got packaged into the phage cells, and upon infection to E. coli K cells, it was able to lyse the E. coli K cells. That is why you got, because it is recombination that has taken place, that is why you got very few plaques with wild type morphology. So the answer here is, which one of the correct conclusions made regarding the R2 mutants A minus B minus from the above experiment? So the correct answer is option 2. The mutation A minus and B minus belong to two different cistrons. That is in the case of experiment 1, and they recombined in the case of experiment 2. So the answer here is option 2. Then next one. The following pedigree represent inheritance of a trait in an extended family. What is the probable mode of inheritance and which individuals conclusively demonstrate this mode of inheritance? Now, if you see, there is father is infected, the son is also infected, daughter is also infected. So it is not any X-linked disease. Here also the parents are not affected but the children are affected. So it is naturally autosomal recessive condition. And there is skipping of generation also. These here, even if the father is affected, the son is not affected, neither the daughter is affected. Here if the mother is affected but the son and daughter are not affected. Here also the parents are not affected but their children are affected. So there is skipping of generation and it is an autosomal recessive condition. So if you see 3, 2, 3 and 4, 1, 2, they conclusively demonstrate the mode of inheritance. That is, it is autosomal recessive. Answer is option 1. Then next one. A chemist synthesized 3 new chemical compounds M1, M2, M3 The compounds were tested for their mutagenic potential and were found to be highly mutagenic. Tests were made to characterize the nature of mutations by allowing the reversion with other mutagens. The following results were obtained. Now M1, so a chemist he synthesized 3 new chemical compounds M1, M2, M3 and these compounds were tested for their mutagenic potential and were found to be highly mutagenic. Then tests were made to characterize the nature of mutation. So you want to know whether it is a transition mutation or a transversion mutation or an insertion or a deletion mutation. So in order to characterize the nature of the mutation, you used certain mutagens which will reverse these mutations and the following results were obtained. So mutation produced by M1. 2 reversed by 2-aminopurine, no. Nitrous acid, no. Hydroxylamine, no. Acridine orange, no. So neither of these could revert the mutation. So nitrous acid, 2-aminopurine, hydroxylamine, they cause transition mutation. And acridine orange, it is meant for insertion, deletion or I can say frame shift mutations. So since M1 is not reverted by any of these, you can say that M1 is because of a transversion mutation. Now coming to M2. Now M2 is reversed by 2-aminopurine and nitrous acid. So it has to be a transition mutation. Why? Because 2-aminopurine and nitrous acid, they cause transition mutation and they can also revert the transition mutation. So M2 is because of a transition mutation. Now M3 is reversed only upon addition of acridine orange. So M3 is reverted back by acridine orange. So it has to be either an insertion or a deletion. So we will see the options. M1 transversion, yes. M2 insertion, no, it is a transition mutation. M1 transition, no. M1 insertion, no. Yes. Then fourth one, M1 transversion, yes, it is a transversion mutation. M2 is a transition mutation, yes, and M3 is an insertion mutation. So answer is option 4. Then next one. A student wrote following statements regarding comparison of restriction fragment length polymorphism, random amplified polymorphic DNA, amplified fragment length polymorphism, and simple sequence repeat techniques used for generating molecular markers in plants. All these techniques can be used for fingerprinting. That is correct. All the above techniques can be used for fingerprinting. Detection of allelic variation can be achieved only by RFLP and SSRS. That is also correct. Use of radioisotopes is required in RFLP and RAPD only. No, that is also not correct. Polymerase chain reaction is required for all technique. RFLP doesn't require PCR. So option D is not correct. So the correct answers are A and B. Answer is option 1. Coming to June 2013 question paper, a chromosome aberration lead to changes in order of genes in a genetic map but does not alter its linkage group. This is due to inversion. Answer is option 4. The concept of recon was proposed by Seymour Benzer by studying recombination between. So he has used T4 bacteriophage. So lysis mutants of bacteriophage T4. Answer is option 1. Then next one. Aspartic acid is specified by a codon GAUGAC. After mutation, aspartic acid is changed to alanine represented by GCX, where X can be AUCG. So, GAU or GAC, it got converted to GCX. So A is a purine, adenine is a purine, and cysteine C is pyrimidine. So a purine is replaced by a pyrimidine. The reversion of the mutation could only be done with reactive oxygen species. The nature of the mutation is considered to be a transversion. Purine is converted to a pyrimidine. So it is a transversion mutation. The next one. A cross is made between two plants with the white flowers. All the F1 progeny had red colored flowers. This is because of. This is the same question that has been repeatedly asked in, I think, 2012. I don't remember the year, the month. But the question has been asked in 2012 as well as in 2017 also they have asked this question, I think. Okay. A cross is made between two plants with white flowers. All the F1 progeny had red flowers. This is because of. So, this is the white flowered parent. Capital A, small b, and small a, small b, small b. Now, in the F1, A and B come together and they complement each other to give rise to what is called as the red flower. So this is an example of complementation. Answer is option 1. Next one. Given below is the result of a complementation test for 6 independent mutants 1 to 6. Plus represent complementation, 0 represent non-complementation. Based on the above, which one of the following conclusion is correct? So when they give a tabular column like this, for complementation, you have to see for the non-complements first and you group them. So first row you can see it is 1, 3 and 6 which are non-complements. 1, 3 and 6 which are non-complements. In the second row, only 2 is non-complement. In the third row, 3 and 6. Okay, 3 and 6 has come in first row also. That is 1, 3 and 6. In the third row also 3 and 6. In the next row, it is 4 and 5. And in the fifth row, it is 0 for 5. And in the sixth row, it is 6. So first you have to group the non-complements. So 1, 3 and 6 have been grouped together in first row. Second row it is 2. So 2 is also taken. 1, 3, 6, 2. Then 3 and 6. In the third row has already been taken in the first row. So that is not taken into grouped here. We have already grouped 3 and 6. Then 4 and 5. Then comes 5 and then comes 6. 5 and 6 have already been grouped. So you have got 3 groups, that is 1, 3, 6, 3, 6 and 4, 5. Now we will read the options. The mutations can be ordered in a single cistron as 1, 3, 5, 2, 4, 6. No, it is not a single cistron, there are 3 cistrons. All mutation belong to single cistron? No. There are 3 cistrons. Mutations 1, 3 and 6 represent 1 cistron. 4 and 5 represent second cistron. 2 represent third cistron. That is correct. There are 3 linkage groups. It is not linkage groups, it is cistrons. So answer is option 3. The next one. In a hospital, 3 babies were mixed up. The blood group of the babies were A, B and AB. In order to identify the parents of the babies, the blood group of the parents were determined. The results obtained were parent set 1, A and AB. Parent set 2, AB and O. Parent set 3, B and AB. Which of the following conclusion can be definitely made? The baby with blood group A is the child of parent set 2. The baby with blood group AB is the child of parent set 1. The baby with blood group B is the child of parent set 3. The parentage of none of the babies can be determined from the given information. Okay, now parent set 1, A and AB. So they can have either AA or AO genotype, and AB will have AB only. So here A, if you take A genotype crossed with AB, you will get A and AB blood groups. If you use AO and cross it with AB, then you will get A, AB, AB. Now parent type 2, AB and O. So it is only you have only two genotypes, AB and O. So you will get either A or B as blood group for your children. Then A and AB, if you take, as said earlier for A here, it can be either BO or BB for the B parent, and AB parent will have only AB genotype. And you can get AB, B, A and B, or B and AB as your offspring blood group. Now what they have told, the baby with blood group A is the child of parent set 2. That can be correct because parent set 2, it can have either A or B only. But parent set 1 and parent set 2 can also have A and A blood group, but you can't definitely say that. You can definitely say for parent set 2. So the baby with blood group A is the child for parent set 2. The next statement, the baby with blood group AB is the child of parent set 1. It can be of parent set 1 or parent set 3. The baby with blood group B is the child of parent set 3. It can be of parent set 1 or parent set 3, not parent set 3 alone. The parentage of none of the babies can be determined from the given information. That is not correct. So the answer here is option 1. The baby with blood group A is the child of parent set 2. The next one. There are two mutant plants. One shows taller phenotype than wild type, whereas the other has the same height as the wild type. So there are two mutant plants. One is taller than the wild type, and whereas the other is the same as that of the wild type. When these two mutations were brought together in a genetic cross, the double mutant displayed even taller phenotype than the tall mutant plant. So what you did was you crossed the two mutants, that is one with taller phenotype than the wild type was crossed with the one having the same height as that of the wild type. They were crossed, and the double mutant, that is the offspring that you got from these mutated crosses, was it displayed even taller phenotype than the tall mutant plant. So this genetic interaction is called as synergetic interaction. Answer is option 3. Coming to December 2013 question paper. A cross is made between pure breeding plant having red colored flowers with a pure breeding plant having white colored flowers. Such a cross is called a monohybrid cross because a pure breeding plant having red flowers, let it be represented by capital R with a pure breeding plant having white colored flowers. So small r. So only two alleles are involved and it is a monohybrid cross. Answer is option 2. Then a cis-trans complementation test is carried out to identify if two mutations are allelic in nature. So a cis-trans complementation test is carried out to check whether the two mutations are allelic or non-allelic in nature. So answer is option 1. The following is the inheritance pattern of a trait under observation. The trait often skips generation. So it is autosomal, not sex-linked, nor sex-linked dominant nor recessive. The number of affected males and females is almost equal. Again, it shows autosomal. The trait is often found in pedigrees with consanguineous marriages. So it is autosomal recessive. So answer is option 1. Then next one. Which of the following representations of chromosomal arrangement in the meiotic anaphase 1 best explains the law of independent assortment? If you see the first option, that itself explains the law of independent assortment. Answer is option 1. Then, the genetic map of 3 genes in Drosophila melanogaster is given below. A cross as given below is made between the individuals of the genotype. The F1 female progeny are test crossed and 1000 progeny are obtained. Assuming that there has been no double crossover, what is the expected number of progeny with the genotypes? Select the set which shows the correct number of expected progeny. So upon crossing these parents, you will get this one as the progeny. A plus B plus C and A B C plus. This will be the progeny. Now that is test crossed. So this is the F1 progeny being test crossed, and you get these 3 progenies, and they have told that there has been no double crossover. So out of this, the C1 will be the parental. A plus B plus C, A, B, C. So that will be the parental type, and the number will be more for C. So you can happily eliminate 3 and 4 where the number is very much less for C. So out of 1 and 2, which is the most correct? So total, you will get only 6 offspring instead of the usual 8, as in double crossover has not taken place. So we will consider only 50% of 1000 as the total progeny. So 50 by 100 into 1000 will be 500. So out of 1 and 2, option 2, if you add together, you will get 500, whereas option 1, if you add everything, you will get 1000. So option 2 is the answer. Answer is option 2. Then next one. The following pedigree shows the inheritance pattern of a rare recessive disorder with complete penetrance. A child from the marriage between individual 2-2 and 2-3 will show the disorder only if parents carry the recessive allele. What is the probability that the child will show the disorder? So here you can see, if this has, 1 minute. Now if this has to be disease, then the parents should be heterozygotes. Similarly here, if this one has to be disease, the parent should be heterozygote. Now probability that now they have asked for 2-2 and 2-3. A child from the marriage between 2-2 and 2-3 will get the disorder if the parents carry the recessive allele. So parents carry the recessive allele. So they are carriers of the recessive allele. So what is the probability that the first parent is a carrier? It is 2/3. Because a cross with AA and capital A small a, you will get offsprings like this, but you will not take small a into consideration for probability. Why? Because the 2 parent and 2-3 parent, they are not deceased. So you will consider only these 3. So out of that, the chances of this particular parent being, that is the first parent or the father, being the carrier of the disease is 2/3. So probability that the second parent is a carrier is also 2/3. Now if parents are carriers, the probability that the child will have the disease is 1/4. So a cross between the carrier parents, you will see like this, and the probability that the child will be deceased is 1/4. 4. Probability that parents will be carriers and the child will be having the disorder is. So multiply all the probabilities. So 2 by 3 into 2 by 3 into 1 by 4, that is 1 by 9. So if you see the options, it is 1 by 9 and the probability of the parents to carry the recessive allele is 2 by 3. So answer is option 1. Now next one. DNA from a strain of bacteria with genotype A plus B plus C plus D plus E plus was isolated and used to transform a strain of bacteria that was A minus, B minus, C minus, D minus, E minus. The transformed cells were tested for the presence of donated genes. The following genes are found to be co-transformed. So A is going with D, B is going with E, C is with D, C is with E also. The order of genes on the bacterial chromosome. So we will start from A. In the options also they have started from A. So A is going with D. So after A it should be D. So that is seen only in case of option 2 and 4. So eliminate 1 and 3. Then D is going with C over here. C, D plus C plus. So D, C. So after D, C is coming. So option 2 is correct. Whereas option 4 is not. But we will see for the others also. After D, it has to be C. Then C is going with E. So after C, it has to be E and E is going with B. So after E, it should be B. So the option 2 is correct. Answer is A, D, C, E, B. Answer is option